of this meeting, and also to all of you for coming. This is what makes this meeting so great. So I will be talking about the role of the mammalian Ino-80 chromatin remodeler in the maintenance of genome integrity during case phase. And as we have heard many times over during these several days, DNA is packaged into nucleosomes, but the enzymes that participate in transcription, replication, and repair have access to it. And that is in large part due to the action of ATP-dependent chromatin remodelers, which slide nucleosomes, evict them, or edit them to maintain regular spacing, to provide space for the access of various factors, and also to maintain specialized chromatin environments where canonical histones are replaced by specialized histone variants. So I'll be talking about the mammalian Ino-80, and in the mammalian Ino-80 class of chromatin remodelers, there are three complexes in mammalian cells characterized by a large insert in the ATPase domain. So these are Ino-80, SR-cap, and TRAP-tip60. And they're involved in various functions in the nucleus. So Ino-80 in particular is involved in transcriptional regulation, various kinds of repair, and the maintenance of the structure of centromeres and telomeres, chromatid cohesion, and chromosome segregation. And we were interested mostly in the role of Ino-80 in replication, and when we started our work, data was coming mostly from yeast, and it was implicating Ino-80 in the maintenance of salt replication forks, in initiation of replication, as well as in some DNA damage tolerance pathways. So we're interested what happens in mammalian cells, mostly because, as you all know, aberrations to the replicative process are now considered to be a driving force in carcinogenesis, as the lesions coming from collapsed forks with time inactivate key tumor suppressors and result in the uncontrolled proliferation of genetically unstable cells. So what is replication stress? Replication stress is any condition that stops or stalls DNA replication, and there are multiple factors that could cause replication stress, including limiting amounts of replication components, DNA lesions, specific secondary structures, as well as replication transcription conflicts. So what happens with a fork, replication fork that encounters an obstacle in its way? It can either be stabilized and maintained in a state that can functionally be resumed, so it continues replicating, or it can be functionally disabled and become the substrate of various nucleases. So we started after this introductory slides, I'll go to our results. So we started by checking the effect of the replication inhibitor hydroxyurea in cells that were knocked down for Ino-80. And what we saw is that the chronogenic survival of these cells was reduced, as well as they accumulated in S phase, indicating problems with replication. So we next checked what happens at the level of single replication forks, and to do this, we applied DNA fiber labeling, which is basically staining the tracks of single replication forks with consecutive labeling with two halogenated nucleosides. So we visualized the fibers by staining for more than one cell cycle with chlorodesoxyuridine, and then we used iododesoxyuridine as a proxy, a short pulse with iododesoxyuridine as a proxy for the speed of the replication forks. And what you can see is that in cells silenced for INO80, you have shorter tracks, suggesting that these cells have trouble with replication elongation. So next we checked what is the origin spacing in these cells, and we saw that the distance between origins is reduced, which is also a property of cells experiencing replication stress. And then we wanted to see what happens when you stop a fork and then you release it. So we first label with the first, our red label, then induced replication stress, and then release the cells, removing the hydroxyurea. So if you get a track with red and green fragments, that means that the fork has continued replicating after replication block. And if you see just a red track, this means that the fork has collapsed. So this is a quantification of the slide I showed you. And as you can see, INO80 depleted cells have increased numbers of discontinued forks, which is indicative of that the forks are less stable there. So something similar happens when you use a different replication inhibitor and when you knock down another subunit of INO80. So the accumulation of dysfunctional forks suggests that these forks need to be repaired by homologous recombination processes. And to follow this, we did immunofluorescence with RAD51, and we saw an increase of RAD51 foci in cells silenced for INO80. And these foci are co-localized with pCNA foci, indicating that the repair process occurs at the sites of collapsed forks. So the take-home message from this part of my talk is that INO80 is necessary for efficient fork elongation, replication elongation, and that INO80 deficient cells accumulate broken forks. So the question is why this happens. And at the time we did this work, there come two papers, one from the Susan Gesser's lab, which showed that INO80 is necessary for the removal of a subset of RNA polymerases from chromatin, and that is necessary during case phase to avoid replication transcription conflicts. And the other paper was coming from the lab of Manolis Papamikos Chronakis, showing that INO80, and INO80 is involved in this process as well as here. And the paper from Manolis showed that INO80 cooperates with the segregase CDC48 to remove permanently arrested and polyubiquitinated RNA PO2s from chromatin and to direct them to be degraded. So what we did, we checked the level of ubiquitinated proteins in cells silenced for different subunits of INO80 complex, and we saw an increase in the total ubiquitinated proteins by a FK2 antibody. And we also saw an increase in ubiquitinated RPB1 subunit of RNA PO2, as well as we had a lower mobility fraction in cells expressing RPB5 GFP following FRAP analysis. So overall, this data indicate that the mechanism described in yeast might be operational in mammalian cells as well. But the question was, to have permanently arrested RNA PO2s, you need a source of transcription stress. People either irradiate with UV or treat with MMS. And in our case, the cells were not treated with anything externally. So this is quite preliminary yet, but we think that the reason for the transcription cells in these cells, in INO80 silent cells, might be R-loops. R-loops are co-transcriptional structures which appear when the nascent RNA hybridizes with the complementary DNA strand, and they're recognized by an antibodies. So we checked if our groups are increased in INO80 silent cells, and we did so an increase. And also there is in these cells there is an increase of the senataxin that is recruited to chromatin, and senataxin is a helicase that unwinds our groups. So we think this might be the reason for what we see. Also, we could rescue the replication, well, partially. We could partially rescue the replication defect in INO80 depleted cells by overexpressing RNase H1, which is also in line with our thinking, but this is still preliminary stuff, so I'll switch to another aspect of mammalian INO80's role in the maintenance of genome stability during case phase, and that is its role in interstrand crosslink repair. So again, by chronogenic survival, we saw that INO80 depleted cells were more sensitive to mitomycin C, and the removal of a crosslinker called 8POP, which is a clickable crosslinker, so you can follow the removal of the lesions by click reaction, is also impaired in these cells. You see that after 48 hours of repair, you have more lesions in cells that are depleted of INO80, which suggested a defect in the Fanconi anemia pathway, which is a fairly complex mechanism for removal of interstrand crosslinks, which is initiated when two replication forks converge on the crosslink. Then FANCM comes to the crosslink, and this nucleates the assembly of Fanconi anemia core complex, which is a large ubiquitin ligase that ubiquitinates FANCD2 and FANCE, and that creates a platform for the recruitment of the nucleases that incise on both sides of the lesion, and the repair is completed by translesion synthesis and homologous recombination. Also, FANCM participates in the activation of ATR signaling in these cells, as it is necessary to accumulate RPA at crosslink stored replication forks, and the activation of ATR leads to phosphorylation of FANCD2 and stimulates its chromatin recruitment, as well as the function of FANCM in a positive feedback loop. So what we did, we checked the accumulation of FANCD2 foci in cells silenced for INO80, and we saw that this was reduced. And also we saw that the monoubiquitination of FANCD2 was also impaired, at least initially, in cells silenced for INO80. So when doing this in live cells and following the accumulation of FANCD2 GFP at a site of microirradiation in cells treated with the photoactivatable crosslinker trioxylin, we saw also an impaired recruitment of FANCD2. And we also saw that key substrates of ATR, namely histone variant H2X and CHEK1 kinase, was impaired in INO80 depleted cells, and these cells failed to stop in S phase after treatment with. I just stopped. Oops. Sorry. Yeah. And yeah, INO80 depleted cells failed to accumulate in S phase. Yeah. Okay. When treated with the crosslinker. So, as this suggested problems with checkpoint activation in INO80 depleted cells, and we followed RPA foci formation in these cells, and the time course shows again a problem with accumulation of RPA. So this could be rescued by overexpression of non-degradable INO80 cells in the ones that were transfected with easy RNase engaged INO80. So it's dependent on INO80. And as I have told you, FunChem is required to accumulate RPA at interstand crosslinks. So we compared the problem with RPA accumulation in INO80 cells and cells depleted for FunChem, and in cells depleted simultaneously of INO80 and FunChem, and we saw that the effects were similar, suggesting that FunChem and INO80 function on the same pathway. So live cell imaging also indicated that FunChem accumulation at sites of interstand crosslinks was impaired in INO80 depleted cells, and we could IP FunChem with an antibody against INO80 and INO80 with an antibody against FunChem when inducing cross-linking damage. So the second part, the conclusions of the second part is that INO80 depleted cells are more sensitive to cross-linkers and are defective in FuncD2 chromatin recruitment. The knockdown of INO80 impairs ATS-dependent signaling in response to interstand crosslinks, and INO80 facilitates FunChem functions and ICLs. And maybe this is the most important of our conclusions, since FunChem is also known to unwind TAR loops in vivo, suggesting that the problems that we saw in replication might be due to impaired, that some event mediated by INO80 on chromatin is not there to recruit FunChem and to act not only on ICLs but also on R loops. So with this, I want to thank everybody involved in this project, my current PhD students, Rusica Hristo and Peter Botev, my ex-PhD student, Evelina Vasilyeva, three very talented master students who, after a brief stint in my laboratory, moved to greener pastures in Austria and Germany, my collaborator, Manolis Papamikos Chronakis, and Dr. Stoyanov Stoyanov as well. So thank you for the funders of this work. Thank you for your patience.
Questions? One question. Yes.
I'm astonished that INO plays such an important role, is not because he cannot play, but there's not any redundancy. So another remodeling, if you kill INO, that this function is, take by another...
it's quite possible. It's quite possible. The effects are not 100%.
Because they are not 100%,
yeah,
yes?
they're not 100%. They could
so
be,
you suspect that it could be some redundancy?
It could be some redundancy,
with, INO.
Yeah. But
and other factors could take his
but
role.
It's almost always the case in cells now. Yeah.
Okay.
More questions? Jose.
For me, but people have been talking a lot about remodelers, and it was very interesting what you present here. But just following up on him, how many other remodelers are out there, and how they work together, and how all these things come, or are just going to go on this road for every? How do we figure out?
There are many, and whether they cooperate or they're redundant, one replacing the other, I would have loved to know, but I don't think that we have the sufficiently sensitive methods to address this.
Just to add to your question about remodelers. Remodelers are in all complexes that deposit histone variant on evict histones. Okay. So they are needed also to destroy chromatin. All the story that you show here, if you have chromatin eviction, histone variants, that are fundamental. They are due also by complexes that have ATPases and that develop force. So they are molecular motors. So as he says, it's really a huge field, and they are involved in all practically processes in biology.
Okay, it's time for a coffee break. We're running just a few minutes behind, but not very many.